Welcome back. Here in this video, I will discuss about the morphology of bacteria. This preliminary information helps you to classify microbes according to their morphology. Not only that, the very first step for the identification of pathogenic bacteria is to observe its morphology. So let's start our discussion. Humans are curious and they want to know the unknown. Antony van Leeuwenhoek was not an exception. He was also curious when for the first time he detected the presence of tiny life under his own handmade microscope. Almost every sample he examined was full of little life and he described them very efficiently. He coined the term animalcules. In 1665, Robert Hooke also observed common bread mold, and after that, scientists from all over the world have discovered microbial life from almost everywhere, like deep ocean, hot spring, permanently frozen area, and even on the cloud. Within 350 years, scientists have identified more than 30,000 different bacterial species. According to them, this number is just the tip of the iceberg. For future microbiologists, there is a huge scope to isolate and identify the rest and find out their unique properties and how those properties could be utilized for human welfare. Although they are very simple, microscopic and single-cell organisms, but they are adapted almost every possible environments because they came almost 3.5 billion years ago and have experienced lot of harsh conditions during that periods. Not only that, they came nearly 1.9 billion years ago than modern eukaryotes and spent more time to evolve. As soon as the microscope and microscopy were developed, it permits us to study microbes in great detail. Like their habitat, they are also unique in terms of size, shape, and arrangements. These three characters are called basic morphology. These basic properties are useful in many aspects, like to classify bacteria into different groups, to identify the genus of the pathogenic microorganism, how they acquire nutrients and multiply, how they communicate with the group, and it gives us a clue regarding how life evolved in this earth. Bacteria are generally 1 to 2 micron in diameter and 5 to 10 micron long. They are too small to see with the unaided human eye. If you compare these three spherical structures, I hope you will understand why they are so small. Among three, the smaller one is with higher surface to volume ratio. And because of that, without any circulatory system, the nutrients are diffused very quickly inside the cell. Without much cytosolic movement, they maintain high metabolic rate, and as a result, they multiply very quickly. Equally, a normal intestinal microbial flora can able to complete one generation within 20 minutes. That means 72 generations within 24 hours. Bacteria are not always small. Look at this bacteria, named Epalopicium fishelsoni. It is 200 to 700 micrometer long and can be seen without microscope. Recently in 1999, another microbes isolated from Namibia called Thiomargarita namibiensis. This bacteria currently holds the world record for largest known bacterium. This microbe ranges from 100 to 300 micron in length and with the largest reported to be 750 micron. On the other hand, nano-bacteria holds the record for the smallest bacteria with a size of 0.08 to 0.5 micron and observed under electron microscope only. They are a little bit larger than the virus. Which factors actually control the cell size? There are several factors that play important role, but nutrient availability is the primary determinant of cell size in many bacteria. Large bacteria are mainly found in certain environments where nutrients are consistently available and concentration are high. It was also observed that E. coli and Bacillus subtilis can increase their cell size as much as 30% under nutrient-rich conditions. Now let's focus onto the bacterial cell shape. Huge variations are also observed in terms of shape, but most of the microorganisms are of two distinct shapes, that is Bacillus and Coccus. Rod-shaped cell is called Bacillus. They are as long as 20 micron and as short as 0.5 micron. Certain bacillus are slender like Salmonella typhi that causes typhoid fever. Others such as the causative agent of anthrax are rectangular with square ends. If the end of the bacillus-shaped bacterium is tapered or pointed, it is referred to as fusiform. When a rod is too short, it is called coccobacillus. Few bacillus species can form resting structure like spore and some are mobile by means of flagella. If the cell is spherical or ball-shaped, the bacterium is called coccus. Cocci can be perfect sphere or oval or bean-shaped or even pointed variants. They are quite small, only 0.5 micron to 1 micron in diameter. You can see the difference between coccus and bacillus from this table. The table is also uploaded in the community section. When the shape of the bacterium is comma-shaped, it is called vibrio. One of the most important members of this group is Vibrio cholerae, the causative agent of cholera. If the bacterium is spiral-shaped, it is called spirillum. It is rigid helix, twisted twice or more along its axis, and another spiral cell with periplasmic flagella is called spirochete. They are more flexible, just like a spring. Some bacteria are pleomorphic. They do not fall within the above mentioned groups. For example, Arthobacter, in presence of nutrients, are bacillus. But when nutrients are depleted, they become spherical. Another nitrogen-fixing soil bacterium called Rhizobium are generally rod-shaped when grow on laboratory medium. But this type of structure called bacteroids are also observed inside the root nodule. Pleomorphism reaches an extreme level in mycoplasma, which entirely lack cell wall and thus display extreme variations in shape. Actinomycetes and nocardia are characterized by filamentous tree-like branching growth, and because of that, initially they were confused with fungi. The greatest diversity in shapes is found mainly in low-nutrient aquatic environment. Cells often have a large surface area that helps them to absorb nutrients more easily. For example, some aquatic bacteria have cytoplasmic extension, giving them a star-like appearance. Square bacterial cells have been found in saltwater lakes. Bacterial cells can also be categorized according to arrangements or style of grouping. The main factors influencing the arrangement of a particular cell type are its pattern of division and how the cells remain attached afterward. The greatest variety in arrangement occurs in coccus. They may exist as singles or in pairs or in tetrads or in irregular clusters or in short or long chains. An even more complex grouping as a cubical packet of 8, 16 or more cells are called sarcina. These different coccal groupings are the result of the cell division in a single plane or in a two perpendicular planes or in several intersecting planes. After division, the daughter cells remain attached. These characteristics arrangements patterns have huge medical importance. When a gram-negative intercellular diplococcus is observed, it could be Neisseria. If gram-positive grape-like arrangements observed, it could be Staphylococcus. If gram-positive short chain observed, it could be Streptococcus. But in order to confirm, some other characteristics are also required. Very few types of arrangements are observed in Bacillus because they divide only in the transverse plane. In this area, they occur either as single cells or as a pair of cells with their ends attached at this position, or as a short chain of several cells like this. A palisade arrangement typical of the Corynebacteria is formed when the cells of a chain remain partially attached by a single hinge region at this end. The cells tend to fold back upon each other, forming a row of cells oriented side by side. Look at this picture. These arrangements, referred to as Chinese letter or picket fence arrangements, are distinctive and helps in the identification of Corynebacterium diphtheria in clinical samples. Spirilla are occasionally found in short chains, but Vibrio and spirochetes rarely remain attached after division. Some prokaryotes characteristically live as multicellular association. For example, Myxobacteria glide over moist surfaces, forming swarms of cells that moves as a pack. When water and nutrients become limiting, the cells come together to form a structure called fruiting body, which is visible to the naked eye. I hope you like this content. If so, press the like button and make your comments. You can also visit the community section of my channel for questions related to this topic. Subscribe my channel if you still don't, and thanks for watching.